Hi everybody. So before we start, any questions from last time? Any questions, comments, anything?
So are we going to discuss the corn experiment?
Yes, we have yet to resolve how can we explain that particular observation. Yes.
Okay.
Yeah. So the idea is to sort of I think I'll be glad if we can finish off the reconciliation between the biometricians and the Mendelians today. And then that sort of completes section one of the course. And then we'll discuss development of modern synthesis of evolutionary biology, which took place in the 1920s, the most part 1930s. And we'll start with an extremely important concept called fitness landscapes, hopefully today. All right. No comments, questions, doubts, concerns? All right, great. Okay. So again, the problem that we have is being able to explain this, that when we were selecting only top 10 oil percentage plants in this cornfield, and then using only those plants to make the next generation seeds, what we saw was a linear increase in oil with time. Perhaps if someone would have asked me, and you can tell me if this agrees with your intuition or not, but if someone had asked me that what would this graph look like, I would have probably said something like this, that when we select the top 10%, then that 10% is primarily due to just variation due to the environment and not so much genetics. So we won't see much of an oil increase for some time. And then one of the plants will pick up a mutation which confers it a higher oil percentage. And then because that particular plant has that mutation which gives it high oil content, it gets selected for the next generation. And that particular mutation then is overrepresented. I mean, when this mutation happened, it was only present in one of the N plants in the field. But then because we use that particular plant to make seeds, its fraction will go up. That particular mutations is present in a greater fraction of the plants, and hence the mean oil content in the population will go up till everybody in the field is carrying the same till everybody in the field is carrying the same mutation, and then the mean oil content freezes for a bit till we wait for one of the existing plants to pick up another mutation which increases oil content further, and so on and so forth. This particular time that's shown here is sort of the waiting time for a new mutation to occur, which gives a higher oil content than what is present in the population at that particular instant. So in some aspect, I would expect this step-like increase in the oil content as we move forward in time. However, that's not at all what was observed in the experiment. Uh, does anybody uh, have a comment regarding this? Does the step-like structure make sense? Guys, anybody?
So I'm not clear why a step-like function will be there. I mean,
Mm-hmm.
if mutation is not taking place,
Mm-hmm.
then uh, even though the normal distribution of height will change, why would it be the step-like increase?
I'm not. Can you say that again?
I mean, if we're selecting the top 10%,
Mm-hmm.
we will see the normal distribution shifting to the right.
Yeah.
Correct.
So
So,
the idea for So I mean, we can represent it. I mean, I have drawn it to be completely flat. You can draw it with a slight positive slope. But the idea behind this step-like intuition is that what is happening here is the top 10% that you see is actually only because of the environmental differences. It has nothing to do with genetics.
Okay.
Does that make sense? Let me pose it another way. So consider this. We discussed different types of selections, right? We discussed direction selection. We discussed a selection type of a selection which bifurcates the population into two. We discussed a type of selection which is called purifying selection. There is one more type of selection which is seen in this particular oil example. And that is that if you have oil content and percentage population here, and you have a distribution like this, when you're selecting top tier, what you're doing is that in the next generation, you are ensuring that only these guys go. There is a certain threshold here to the left of which nobody makes it to the next generation. And this type of a selection is what is called as truncation selection. That selection truncates, selection basically lays down a marker here and everybody to the left of it never makes it to the next generation. And only these guys are represented in the next population, next generation. This is an example of truncation selection, and this is most commonly used in artificial selection experiments. I mean, nature obviously rarely would put a truncation selection where a hard marker is laid down, where only one side of the marker makes it to the next population. But let me ask you this, that if you were to take these 10% and mate them, right? What is the expectation of the offsprings that you see in the next generation? And I want to see show of hands, but let me lay down the 4 options. The first option is that, so this line should be vertical, but you get the picture. Basically, if I produce offsprings from here, I would get a distribution between these 2 windows. In the next generation, this is the window that I see. Option B is that if I were to mate all of these guys, I would get back the original distribution, same mean, no difference in the mean, which basically means that the variation that was seen in oil content in this particular population was all because of noise or environmental factor. Genetics didn't have anything to do with it, because when you mated these guys, it was brought back to the original distribution. Option C is that it will be a new normal distribution between these two, but you will have some individuals which lie outside of the window from which this was chosen. Some guys will go even to the right of the maximum oil content, and some guys will go even to the left of these guys. That's option C. And option D is that let me make this smaller. Sorry, I should have made this more carefully. Option D is that the distribution, let me make it smaller still. Option D is that the distribution has, so this leftmost line is the mean of the original population. And option D is that the new distribution is slightly to the right of the original distribution, but not as far as the mean of the 10% that were chosen. So the population has moved slightly to the right. And now I would like you to comment, show of hands who think that it will be A. Is the question clear?
Yes, sir.
All right. Show of hands who think it will be A. 1%. All right. B. When somebody votes for B, essentially what you're saying is that genetics has no factor in dictating oil content. It's all environment and noise. So no one's saying B. Fine. C. All right, lots of people with C. Cool. And D. All right. Why C?
Because
So just one second. So nobody said B, right? So which means that none of us thinks that oil content is a trait which is independent of genetics. So that's fine. Rishi said A, and I understand the intuition behind it, because now you're only choosing individuals between a certain range. You're going to get offsprings in that certain range. Yeah, C, go ahead. Who was that? I'm sorry, I missed that.
It was me. So, for me, I chose because in that certain range of corn oil content, the mean would be what the mean of the range is, and the variation would increase because of other environmental factors adding on to the new offsprings. Hence, the
Okay.
variation will not be the same or decrease; it will increase.
So, but why should this variation be greater than what was seen before? Why should the variance of this distribution be such that the range of the phenotype, the range of the oil content, is greater than what was seen in the parents of these guys?
Um,
That's okay. That's fine. I mean, I think that's not a trivial question. So I understand the logic. Anybody else with C who wants to add to what Akansha said?
Yeah, you know,
Yeah, Anirudh. Yeah,
yeah,
you
so
said
I
C,
think,
did you?
yeah, I didn't raise my hand, but I think it's C2.
Mm-hmm.
I think I mentioned this last time, but it has to do with recombination, which increases the variation frequency in each generation in sexual selection. So that
Why would it do that?
because it's first of all, recombination is a random process, and the rate of recombination is much more larger than the rate of mutation. So it's more frequent that you would get a variation out of recombination than a mutation. And that's why you can see a higher amount of variation in the next generation.
Right.
Yeah.
Yes. Okay. Anybody else? Anything else has to say? Anything? Yeah. So it's essentially what Anirudh has just said. So for those of you who are not biologists, let's go through these couple of slides. Essentially, what is happening is that, I mean, we can think of our parents. One parent has these two copies of the chromosome. One of the copies, I would like you to be familiar with the words allele and polymorphism, what we discussed last time around. So one of the copies of the chromosome has genes, alleles A1, B1, C1, and D1 at these 4 different positions on the chromosome. And for this particular individual, the other copy of the chromosome has alleles A1, which is the same as the allele present on the other chromosome. At this particular position, one of the chromosomes has B1, this chromosome has B2, then C1 and C2, and D1 and D2. So that is the genetic composition of individual 1. Similarly, the other parent has A1, A3, B2, B3, C1, C3, and D3 and D1. Now, when it comes to these two individuals mating and producing an offspring, this individual has to pass down one copy of the chromosome. And we briefly discussed it in one of the lectures that the copy of the chromosome that this guy will pass down is not going to be one of the two. It's going to be a mishmash between the two. So it might copy chromosome 1 up to, well, it might make a copy of this chromosome up until this point, but then switch to this one. Then it might copy these two and then switch back to this one. So what you will get is A1, B2, C2, D1 as the one copy of this chromosome, which gets passed on to the offspring. And the same happens here. This happens this is done via sperm and egg in human beings. So you might have, if the original parent's fitness was F1 and F2, it's not necessary that they have to contribute. It's not, I mean, it doesn't work in the way that they only contribute one of the chromosomes. If this guy contributed one of the two chromosomes as it is, then there were two possibilities of this. And same with this, there were two possibilities of donating one of the chromosomes in this individual. So the total number of progeny that you will see would be 4, depending on which copy of the chromosome was taken from here, which copy was taken from here. But because of this process of hopping between chromosomes and making a hybrid chromosome, this is called recombination. What you might get in the offspring, I'm shown just one particular manifestation, that A1, B2, C1, D2. Basically what that means is it copied this one, then it switched on to this one, B2, then it switched back to C1, and then it switched back to D2. So you get this sort of a hybrid chromosome, which came from this parent. And from this parent, you got A3, B3, C1, and D1, which means A3, B3, C1, and then it switches back D1. And because of this, you get an offspring, which is fitness F3. So, and obviously there are so many other combinations also. And then you have to think that, for instance, in human beings, this is just one pair of chromosomes. So this could be, let's say, chromosome 1, but we have 21 other chromosomes in addition to X and Y chromosome. So there are 21 other pairs in which the same process is being repeated, and hence the amount of variation that you can generate in the next generation because of this process of recombination is just astronomical. So you are not, as Anirudh said, you are not in sexual reproduction because of this process of recombination. The amount of variation that you generate without acquisition of any new mutations is just astronomical. Okay, so what, again, sort of summarizing the same thing, that new fitness levels are accessed without acquisition of new mutations. And because this is because repackaging of genes because of recombination and sexual reproduction, and sexual recombination takes place. And as a result of this, greater genetic diversity is generated in each generation via sexual reproduction as compared to asexual reproduction. And this is the fundamental difference that I was talking about last time around when I said that we will come back to the continuous versus discontinuous debate. But before that, we have to realize a fundamental difference in the way in which diversity is generated in an asexual population compared to a sexual population. Remember, in an asexual population, we said diversity was generated via acquisition of new mutations. There are other mechanisms also, but they're not as prevalent as something that we've just discussed in sexual organisms. Any questions with this?
So,
Yeah.
so DNA recombination takes place in plants as well, right?
Yes.
In self-pollination mode.
Recombination will take place whether it's being self-pollinated or crossed with another individual. Recombination will take place, but when you're mating with self, it's called selfing. Okay, when selfing is happening, what is happening is that your chances. So you can think of it like this: when selfing is happening, this is parent 1, and this is also parent 2. Does that make sense?
Yeah.
Right? So both the parents, the original pair of chromosomes in which recombination is going to take place is the same. So you can basically just copy this and put it here, and then hence you get the 2 parents. Does that make sense?
Yeah. In that case, also variation will happen, right?
Variation will happen, but it will happen to a lesser extent. Right? I mean, it's a simple question of permutation combinations, and that reduces when selfing is happening.
Okay.
Right? And the bigger issue with selfing is, and it's a big literature. The bigger issue with selfing is that suppose I am an individual and I am carrying, let's go back to this B2 allele is extremely bad. Okay. So I am only, B2 doesn't function at all. And this particular locus serves a very important function in the cellular being. So if B2 is bad, I am still able to survive because I have a functional B1, right? But if I am reproducing via selfing, if I mate with myself, there is a 25% chance that my offspring will have B2 on both the chromosomes. My pair will have B2 and B2. Does that make sense?
Yeah.
Right?
Yeah.
And as a result of that, my offspring might not be able to survive depending on how critical is B1's presence to survival of the offspring. So there is a 25% chance that my offspring might not make it. And for this reason, selfing is considered a bad idea, which is essentially what you are doing when you are mating within families. That if your family tree happens to have some sort of a genetic disorder, you are increasing the likelihood of producing an offspring which is carrying both the alleles which are not functional or suboptimal, which is what we saw in the Habsburg family tree, that there was so much not selfing. Obviously, that doesn't happen in human beings, but there was so much inbreeding in that population that with each inbreeding you are increasing the chances of producing an offspring which might have two alleles at a locus which are non-functional. Does that make sense?
Yes, sir.
Right. Cool. One
Sir,
more. Yeah. Go ahead.
coming to the distribution part, can you show that graph? That
Yeah.
plot?
Yeah. Yeah. Tell me.
In the original one, that particular recombination is not present. So that much oil content, the probability of producing that much oil content is not there in the original plot. But when
No. I didn't understand you. Say that again.
okay, there's a cutoff, right? Like the last line is going, but in the C option, the distribution of that part is seen, like the probability is seen, but in the original plot, probability is 0 in that region.
In this region, you mean?
In the last part, I mean, it is exceeding that line, right? That cutoff
This
line
one?
is
This
not
one?
present in the original one. Yeah, no, in the C1 last line.
This one?
Yeah, that region.
This
That
region.
small region that is not present in the original distribution.
Exactly. That is why I have option C. Yes.
Yeah, why is it so? Because that that recombinant might also be present in that whole distribution, right?
This distribution.
Yeah.
So that is exactly what we have to solve. But no, let me come back to that. Okay, I just want to make one small comment before we come back to the oil problem. Okay.
Yes.
So what we've been saying is that recombination does this repackaging business. You have alleles, and these alleles get repackaged differently to create genetic diversity. We can think of an individual as a set of alleles that he or she is carrying. Now, what recombination does is produce a set of individuals which is carrying a unique set of alleles based on how recombination happened between the parent's chromosome. But recombination can do one more thing. Recombination can produce new alleles that did not even exist in the original population. And that can be seen. Imagine if this is being copied. This guy is undergoing meiosis and producing a sperm or an egg. Now it might happen that it copies this part of chromosome 1, and while it's in the middle of this gene B1, it switches to this part. If that switch takes place in the middle of a gene, then what it passes on to the next generation carries the starting part of B1 and ending part of B2. Now this new B allele is neither B1 nor B2. So you have created a new allele only by recombination, and you didn't need to resort to mutation to create a new allele. And this new allele is obviously going to have a newer phenotypic manifestation in the individual. Does that make sense, that how recombination can lead to newer alleles too?
Yeah.
All right.
So, sir, is recombination the answer to what Darwin was looking for when he was thinking about blending and how blending decreases variation, but something is there which increases variation? So, is that recombination?
Yes, it could be. But before we get to that, let me go back to the oil problem now, and sort of the question that Namrata raised. That why is it that, so how do we explain, okay, maybe not here. Let me go back to this graph now. Now, in the light of this, how would you explain this linear increase?
So in each generation, now we are seeing a little deviation from the variance is increasing in each generation. That's why we are seeing the
Variance is
deviation.
increasing. Variance ka, we don't know anything about variance, right?
Okay,
Variance
okay.
in what?
So the mean keeps increasing, right?
Mean is increasing, but that is an observation that we have seen from the graph. How do we explain that observation now?
Sir, so the rightmost value or the biggest value is now constantly increasing
Why?
because of the variation, that added variation on
Which
the
added
right side
variation?
of the,
Which added
Due
variation?
to recombination.
yeah, due to recombination.
What is recombination doing?
It is increasing the number of variations in the higher oil content side, and that would mean that the number of population that we thought previously was only constrained by the maximum of the first generation is not the case now. The maximum is also shifting, and the mean is increasing. Hence, we can see the trend.
Anybody else? I mean, you guys are essentially right, but I just want a more clearer description of what's happening. Okay. So, Namrata, what is happening is the following. C is the correct answer, right? It doesn't mean that C is the correct answer in every single case, but in this case, it happens to be true. What is happening is that these guys are carrying some alleles and they have a physical manifestation in terms of exhibiting this range of oil content among the plants. When we go to the next generation, the alleles that are present in these plants are present. Each plant is carrying a certain set of alleles. When we go to the next generation, recombination happens and newer combinations are generated. These newer combinations were not present in this particular population. As a result of that, some of these newer combinations are going to give me an oil content which is actually lower than what was seen, but some are going to give me an oil content which was higher than what was seen in the previous generation. These combinations are perhaps those which were not present in this particular population, and they were created by recombination. Either because recombination created alleles which didn't exist in the previous population, or because recombination led to packaging of existing alleles into sets which did not exist in the parent generation. Now, once I get this, I will place my threshold at 10% and do the same with these guys. And the same story will keep on. Now I'm repackaging the alleles which constitute this particular bunch of individuals. And I will again get a distribution of plants here, some of which will exhibit a phenotype beyond the currently one, and some of them will exhibit a phenotype which is not present in these 10%, and so on and so forth. And it will keep on going forward. Does that answer your question, Namrata?
Yes, sir. So then coming to the whole population, like not selecting only the higher 10%, will it happen in the next generation also? Means the variation keeps on increasing.
If there was no selection?
Yeah.
This is what's happening if you apply no selection, the dotted line.
Okay.
Yeah.
Like, why is it so? Because a recombination can happen, and like new features can, like higher oil producing feature or lower oil producing can come into picture, right?
Correct. But now selection has nothing to do with them. Selection now has who produces. Oh, I'm sorry. Selection now has to do with who's producing more seeds. So if you left everything on its own, you will get a particular normal distribution, right? It doesn't mean that you can't go beyond that normal distribution. You can, but the frequency of that happening is very small unless you are selecting for it, right? Like the example that we discussed last time around, that one when we grow a bacterium in the absence of antibiotic, and we start with one bacterium and we end up with 10 part 12 bacteria, is it possible that an individual will be there which is resistant to antibiotics? Yes, it's very possible that individual will be there, but unless we apply selection for it, we will never find that one individual from the 10 part 12 bacteria that are present in the flask. Does that make sense?
Okay, the probability is very, very, very less.
Is extremely small. And when we are talking a normal distribution, we are essentially talking just probability. It's a probability distribution, right?
Okay.
So when we have a finite population, which in this case we do because the plot of the land in which this experiment is done is fixed. When we have a finite population, the chances that without selection, that particular trait will spread in the population. So what you're asking is, can we get a scenario where we will have oil content change in the population without applying selection on it? Is actually an interesting question. And this concept in evolution, where we have chance events dictating fate of populations, is called drift. That process is called drift. And we'll spend quite a bit of time talking about drift, that how can chance events dictate evolutionary trajectories when something happens despite there being no selection for that particular phenotype. So we'll discuss that, but the chances of that happening are extremely small.
Okay.
Yeah. And it becomes higher, those chances increase the smaller your population gets. Chance events become more dominant in very small populations as compared to large scale. But I mean, we're going to have a whole new discussion on drift in a few weeks' time. Any other questions, comments?
So I had a question.
Yeah.
So my question actually is, does the sloppy DNA recombination the cause of mutations?
I'm sorry, say that again. Does that what? Sloppy, did you say?
Yeah, I mean, when DNA recombination breaks an allele from the middle of it and forms a new allele, this forming of new allele, is it termed as mutation, or is something else responsible for mutation?
Not really. When you say mutation, generally what you mean by it is error in copying. Okay. I guess Yash got disconnected. We'll just yeah. So generally not. When you say use the word mutation, you're generally referring to an error in the copying mechanism. Recombination is not an error in the copying mechanism. The replication of the DNA is extremely faithful. It's copying whatever it's reading. So recombination is a mechanism by which variation is being generated independent of mutations. Okay.
Yeah.
We are still not quite done. There is one more aspect that we need to realize before we can explain this. Okay. Imagine that this trait was being governed by only one particular locus. So when we are talking DNA of an organism, and let's say our chromosome number 1 has these 4 different positions, each one of these is called a locus. When we are talking of a particular location on the DNA, it's called locus, and obviously the plural is loci. So let's imagine that oil content is a trait which is decided by only one locus, okay, what do you expect the graph to look like in that case? Is the question clear?
It will plateau out
Why is that?
eventually. Like, there are many alleles whose locus space supposedly, but after some time you'll get the best combination of alleles. And
Right.
once you start selecting for those two best alleles on your two copies of the chromosome, uske baad it should not increase further.
Absolutely. Yes. So think of it like this, that when we are talking of oil as a trait, and oil is only dictated by this particular locus. So everything else, these three loci, A, C, and D, and every other loci on every other pair of chromosome is irrelevant as far as oil is concerned. That means the relevant alleles for us are B1, B2, and B3 only. And one of them is going to lead to maximum oil content among the three of us, not three of us, three of them. And what is going to happen is that, let's say B3 was the best one. B3 gives you the highest oil content. So all the plants that had B3 would be towards the right in the normal distribution, and I would be selecting them, right? And very quickly, what would happen is that B1 and B2 would get eliminated. And then how would oil content go up? Would I see a further increase then?
No, you won't see it, right? Once it's in like the homozygous dominant sort of condition.
Yeah. So once
There
I get
wouldn't be any
into exactly. So once I get into the situation where once we get into a situation where every individual is carrying a B3 allele on both the copies of the chromosome, then you cannot have further variation being created except for newer mutations to occur. And when we get into that scenario, this is the dynamics that I expect. That this is the period when B3 spread into the population. Now everybody is carrying B3 on both copies of chromosome. Now you have to wait for a long time before one of the B3s becomes a B4, and that B4 is of a kind which actually supports a higher oil content. Then I will start selecting B4 in my next generation's population, and now everybody is carrying B4. And now I'm waiting for a mutation B5 to occur, and so on and so forth. Does this make sense? Yes, no, repeat.
Yes,
Everybody?
So,
Yeah.
but the plateau period would not be same in each case, right?
It would not be same. Perfectly, absolutely. I mean, mutations are a random process, and in one of the lectures, when we do quantitative aspects of this, we will actually calculate that what is a waiting time for a mutation. So waiting time for a mutation is obviously going to be variable in every single case. But what is the mean and what are its variance features? We will do that calculation in one of the classes. But in essentially in qualitative nature, does that graph make sense?
Yes, sir.
Okay. So now we have run into a problem that if a trait was decided by one locus, then the physical manifestation of that trait should have been a step-like manifestation. What we see from the Illinois experiment is that it's a continuously increasing graph. What does that tell me about the trait of oil content and the number of loci that are controlling it?
So it might be multiple loci controlling.
Yeah, Sangamitra?
It should be more than one. So, but is it like most, like, other than this in general, like all the traits that we have, like we are having recombination, but like it's kind of constant, right?
What is kind of constant?
Like our genotypes.
Well, as we discussed last time, that the number of nucleotide differences between you and me are of the order of 4 to 5 million. Right?
Yeah,
So there is
it's like
quite
a
a bit
general
of,
polymorphism that exists like that.
what was that?
Like it's like a general polymorphism that exists. So
Correct.
yeah, it doesn't flatten out basically.
Correct. Exactly. So what is now thought about this experiment is that what that linear increase tells me is the fact that oil content as a trait is controlled by many loci, which is why, I mean, it's exactly a century and a quarter now. After 125 years, oil content is still increasing linearly, which means recombination is actually still repackaging these genes, these genomes in a fashion such that newer alleles are being, newer allele combinations are being formed. Maybe newer alleles are also being formed. And all of this, what this tells us is that oil content is a trait which is decided by at least uh, dozens of different loci which are present on the genome. And what should be the time frame of this event? I mean, eventually it should saturate, right? So what is now thought is that newer mutations happening is actually so insignificant in this particular experiment that even if there were no mutations occurring, the trajectory of oil content would be more or less the same as what we have seen in this experiment. Now, theory tells us that. And sort of the next section in the course is going to be talking about that can we make predictions about these dynamics of evolution, that how long is something going to take. Like Sakshi mentioned that the waiting time for a new mutation shouldn't be the same in every case. I mean, the cartoon I drew had the same, which is obviously wrong. But it will, the waiting time is going to have some statistical features. If we have a beneficial mutation happening which gives you an oil content which is 3% more than everybody, how long will it be then that that particular allele spreads into the population, because it is being selected in every single generation, and we get to a situation where every individual is carrying that particular allele? How many generations is that going to take? What happens to the fate of mutations which actually decrease oil content, right? All these, Namrata mentioned the chance of drift, or the chance, the probability of chance events dictating evolutionary trajectories. These are some of the issues that we'll be talking about in the next section now. As far as biometricians and Mendelians are concerned, Mendelians insisted that it happens, selection acts on discontinuous variation because Mendelian genetics is correct. What they did not realize is the fact that Mendelian genetics, when coupled over many loci, if each one of these loci was contributing independently of each other to a particular trait, then Mendelian inheritance is actually consistent with a normal distribution of the trait. Because you are going to see, every time recombination happens, you're going to see slightly different repackaging take place. And you will see phenotype being extended to the right, as in this case, just a little bit. So it would, even though Mendelian inheritance was at the play, it was dictating the dynamics and the phenotype of the population, but it didn't manifest as discontinuous variation, because traits like height, weight, oil content, these are called quantitative traits. And the number of loci which dictate these traits, which is called quantitative trait loci, actually runs into hundreds, thousands, hundreds of thousands, as in some of the cases that we know now. And this is sort of a biggish area now that many people are working in, trying to decipher the number of loci that dictate a trait in a particular population. Okay. So one of the essential elements to us observing that linear increase in oil is the fact that many loci dictate the trait. One more thing needs to be satisfied before we see that. Recombination will only lead to newer combinations if there was enough variation in the population that I was starting with. If all the individuals had A1 at this allele, B1 at this allele, C1 at this allele, recombination cannot do anything, right? Those of you who worked with bacteria, we start a culture with what is called an isogenic culture. That means every individual has the same DNA sequence. Every individual that I'm starting a culture with has the same DNA sequence. Now imagine if this plant experiment if this corn experiment was done with a bunch of individual plants which had the exact sequence, what would the experimental outlook look like? Is the question clear?
Yes, sir.
If every individual was carrying the exact same sequence in this experiment when the experiment was started off, in other words, all the alleles in the population were exactly the same, what would be the outcome of that experiment?
It would have been like waiting for the mutation until and unless it would have been the same.
Yeah, so then you again run into the graph. Yeah, Sangamitra, go ahead.
I think there will be a certain, till a certain level, there will be recombination, and then after that, there will be a waiting period for mutation.
Yeah, but recombination now is only going to help if recombination is creating new alleles.
Yeah.
Right? Because recombination now is, if it's just repackaging alleles, it's not going to create a newer genotype because there is no variation in the population. Right?
Sir?
Yeah.
In that case, you won't be able to find the top 10%, right? Because everything will have the same amount of oil.
It's the progeny from where we are getting it, so we will, I guess.
So, if you had an identical twin, Pavitra, would your identical twin be the exact same features as you?
Yeah, probably.
Exact same features.
No,
right? There'll be slight variation. Why would that variation be?
I don't know.
So the variation will be because of the environment, right? Maybe she will eat better than you, or sleep better, or exercise better or worse than you, and so on and so forth. So every phenotype, which in this case is oil percentage, is dictated by two components. One is the genotype, and the other is the environment that this thing is grown in. Suppose I don't know this for a fact, but let's say plants produce very low percent oil in corn if they are grown in very dry conditions. Okay, now if I take the exact same seed, identical sequence, I plant one in high moisture where I water it properly, and the other one where I keep it dry, right? Will there be variation in the phenotype?
Yeah.
Right, there will be variation. And what is causing this variation? What is causing this variation is the environment that this experiment was done in, right? So even when I grow these genetically identical plants in the same field, and I might try to keep the environment same as well, the environment will not be the same. Some of them will get more sunlight than the others. Some of them are exposed to wind more than the others. Some of them, the soil has a certain chemistry which at other parts of the field it's not there. So all these factors are what are called environmental factors, right?
Okay.
And this environmental factor, even if my population was genetically identical, it will give me some variation. Now that variation may not be as broad as this, that variation might be smaller in that case. But then we are talking just a quantitative amount of variation, right? Imagine all this variation was due to environment. Genotype had no role in it. All these were genetically identical. Then when you choose the top 10%, in the next generation, when you plant these guys, you should get the exact same distribution again. Does that make sense?
Yes, sir. Yes.
Right? Because any variation was because of chance event. Right? A good way to understand this is that imagine a class of 100 students. Okay. Now, I give a quiz where there are 100 objective type questions, and for each question there is a true and a false option, right? Let us assume that no student knows any of the answers, right? And everybody answers all 100 questions purely based on guess. They have no idea whatsoever. What would be the mean score, Pavitra?
So everybody is guessing it, right?
Everybody is guessing every question.
It could be the 50% of it.
50%, right? The mean score would be 50% because everybody, but will all students get 50%?
No.
No, right? Some will get less, some will get more, right? Now, let's imagine that I pick the top 10%. Okay.
Yes.
And to these top 10%, I give another quiz where there are again 100 questions, again true false type, and again none of them know any answer whatsoever. What will be the mean of this new quiz now?
Or it'll be more than 50.
Why will it be more than 50?
Okay, no, it'll still be 50. It'll still be 50.
It will still be 50 because they're again guessing, right?
Yeah.
Because whatever variation was there in the original test was because of chance.
Got it.
Right?
Yes.
Right? It's the same thing happening here. That in this population, these top 10% individuals are exhibiting this trait because they happen to be maybe in the right sunlight, or the soil chemistry was chance events, nothing to do with genotype. So when I take these individuals and I think that these are high oil producing plants, and I plant them all over the field, now the same variation that was affecting the entire population is now going to affect the seeds of these plants when planted all over the field. So when I plant these, and if the trait is being dictated only by environment and not by genetics, or genetically they're all identical, then the distribution that I'm going to get in the next generation will be identical to the one that I just saw. Does that make sense?
Yes, sir. Yes.
Yeah, so a trait is dictated, any trait is dictated by environment plus genotype, right? So if we have a population where every individual is the same, then the dynamics of the oil experiment in Illinois will not be the same, because recombination cannot create new strains because every individual is the same. Repackaging of existing alleles is not going to help because every individual is carrying the same alleles. And hence, what will happen is that we have to wait for new mutations to arise for oil content to change. And that will lead us to a physical manifestation such as this. Does everybody find that agreeable?
So it
Yes,
would be
sir.
like selfing.
I'm sorry, one at a time. Akansh?
I said it would be like selfing, right?
It would be like selfing, yes.
Yeah, okay.
Yeah, Sakshi?
No, I was saying yes, it makes sense.
Okay, okay. Any questions with this?
So I had a question.
Mm-hmm.
So how likely is a new allele to be produced in the recombination?
In sexual organisms, not very likely. And that is because on one chromosome, so we have 23 pairs, on one chromosome, recombination only happens 2 or 3 times. Okay. And if we look at human beings as an example, if on one chromosome recombination is happening 2 or 3 times, a new allele is likely to be produced when a recombination event happens in the middle of a gene. I mean, not middle, but anywhere inside the gene. But if we look at the entire DNA of human beings, more than 98% of the DNA is actually does not encode for genes. And by genes, that means it doesn't get translated to amino acid chain, and then more than 98% of the DNA is just what we call intergenic DNA, which is the region of DNA in between genes. Now that probably has some role to play in dictating the physiology of the organisms, but we don't know for sure. So most likely recombination is not going to create new alleles. It's just going to happen in the regions in between genes, just because of the fact that there is much more of DNA which is not coding than coding regions. Does that make sense?
Yes.
Last comment before we move ahead is that this kind of a variation that existed in the population. So now we know, looking at the Illinois experiment, that the alleles present in the population, different individuals must have carried different alleles for this kind of trajectory to be observed. This variation that must have existed in the population before the experiment was started is called standing genetic variation in a population. I thought I had wrote it down. Yeah. This kind of variation is called standing genetic diversity, or variation that existed in the population that before at the start of experiment. And this diversity allows for newer combination of alleles to be packed, and then selection can act on this newer diversity. Any questions with any of this? So now, as long as a trait is dictated by many alleles, you are going to have continuous variation take place. Even though dictating that continuous variation is Mendelian genetics. And that was the reconciliation between biometricians and Mendelians. And this was again proposed much before alleles were known, DNA was known, genes were known. And this was a seminal paper written by Ronald Fisher in 1988. Extremely hard paper to write. Read. I'm sure write too. But sort of, it's, I think, easier to understand in the backdrop of what we know now, and reconcile this debate between biometricians and Mendelians. If the trait is being dictated by fewer number of loci, then the dynamics of evolution would be very different than what we've seen from the corn oil experiment. Okay. All right. We will now move to something called a fitness landscape. Essentially, now this is the beginning of. So after this debate, it was primarily 3 people who laid the foundation of what is called modern synthesis in evolutionary biology. And this modern synthesis essentially encompasses every aspect of evolution as we see it now. And what we will see is, while we discuss concepts from the time, so the period of the timeline that we are talking about now is 1920s and 30s. That's when the foundations of modern evolutionary theory was laid down. And what we will do is discuss concepts from the same time. But remember, all of this was done when no details of biology were known. And what is astonishing, at least to me, is that while all of these things were discovered back then, it's only now, particularly in the last 10 years or so, that aspects of these concepts are being experimentally explored now, because the technology now allows us to explore some of these concepts that were proposed back then. But these concepts were, I guess what I'm trying to say is that these concepts were just so far ahead of their times that we are only just scratching the surfaces of these concepts when it comes to experimentally testing these ideas in lab. And the modern synthesis in 20s and 30s was essentially a product of work of three men. One of them is Ronald Fisher, who wrote the 1918 paper. The other one was JBS Haldane, who incidentally had a major falling out with the British government because of their participation in the World War II, and eventually was in love with socialism and sort of saw his future and ideology aligned with the idea of India in the time of Nehru, and hence moved to Bengal soon after India's independence. So that's a pretty nice story to read in its own right. But what we're going to start off with is a contribution by the third person in this trio, called Sewall Wright, who was an American geneticist. And what we would like to do is visualize evolution take place in a geometric sense. Okay, and Sewall Wright gave us a tool in a paper that was published in 1932. He gave us a geometric tool in which to visualize evolution take place. And what we will do next is sort of describe the concept of this geometric tool, which is called a fitness landscape. And from there on, we will read the two papers that I shared with you. It's 2006 and 2011. And these were sort of the first attempts to experimentally decipher what Wright was talking about. And so that's an idea of sort of how far ahead Wright was of his time. But today and probably Wednesday also, we will be discussing at a conceptual level, the construct that Wright was talking about. And I know I wrote the submission of these two papers was due on Friday, but I mean, it looks as though our discussion on fitness landscapes is going to extend into Wednesday, and I don't want summaries to be due just 2 days after our discussion. So instead of Friday, let's make it the Wednesday afterwards, as far as the submission is concerned. All right, so we'll look at fitness landscapes now. A few questions. Imagine a sequence of DNA of length L nucleotides. So you have a sequence of length L. L in human beings' case is obviously 3 into 10 power 9. How many such unique sequences are possible? I mean, it's written here, but does that make sense?
Yes.
Yeah.
Anybody, if it doesn't make sense? Everybody's okay with it? All right, cool, great. So in order to understand this, let us number them. Let us number these sequences as 1, 2, 3, ij, going all the way to 4 to the power L. Okay. Remember, if we are talking L as the genome of the organism, we are talking an astronomical number. I mean, there aren't enough atoms in the universe, 4 to the power L, where L is 3 into 10 power 9. Or even a bacterium where L is of the order of 10 power 6. 4 raised to power 10 power 6 is more atoms than what is thought to exist in the universe. So this is an astronomical number. So it's a geometrical concept. It's an abstract concept. Let us number them. So we now we have numbered all these sequences. So number 1 is a sequence, number 2 is a sequence, and so on and so forth, going all the way to 4 to the power. We'll represent each one of these sequences as a node. So now we will draw a graph, graph in the sense of what computer scientists call graph. So we will draw this graph of 1 to ij going all the way to 4 to the power L, and we have these nodes. So far so good?
Yes, sir.
Next, we have to discuss that for given any two sequences, there is a concept called Hamming distance. Hamming distance between any two sequences is simply the number of positions. So Hamming distance between two sequences is only defined if those two sequences are of the same length. In our case, all these sequences are of length L, hence we can compare the Hamming distance between them. Hamming distance is simply the number of positions where the two sequences are different from each other. So if I'm comparing sequence 1 and 2, the Hamming distance between them is 1 because these two sequences are dissimilar here and they are identical everywhere else. Hence Hamming distance between 1 and 2, which is written as H12, is equal to 1, which is the number of bases which are different in the two sequences. Similarly, sequences 3 and 4, Hamming distance is 2 because now we have two positions at which the bases are different in the two sequence. Is this clear, Hamming distance?
Yes.
All right, great. Now, remember we have the 4 to the power L nodes where each node represents a sequence of length L. Now in this graph, we will connect nodes I and J with an edge if Hij is equal to 1. Okay, which means that if there is only one nucleotide difference between 1 and 2, we will connect them with a edge. There is only one nucleotide difference between I and J, and we can connect it with an edge. Now we get this two-dimensional graph, and again, from the context of an organism, this is an astronomical figure, which is simply not possible to be made. We get this graph now, which is called a sequence space. Right? This particular graph is called the sequence space of the organism. Next question: how many neighbors, and you are a neighbor if you are connected by an edge? How many neighbors does each node have?
2.
No, not even close. Think about it. You are connected with another node if the Hamming distance between you and that node is equal to 1. So the question is that how many other nodes are there with which you have Hamming distance equal to 1?
It can be 0 or many.
Oh, one at a time, Sangamitra.
It can be 0 or it can be like more than one. Like it's not definite, right?
It is definite. Because remember, 4 to the power L is every possible sequence. This is a theoretical construct, right? 4 to the power L is every possible sequence. So all possible sequences of length L are there. And among all these sequences, how many are at a Hamming distance equal to 1 from you?
L.
L minus 1.
L minus 1, yeah. So,
L minus 1, yeah. Okay.
Okay, others? Let me say L minus 1 is wrong.
So 4 to the power
4
L
raised to L minus.
I'm sorry, I got distracted. Say that again.
4 to the power L minus 1. L minus 1 in the experiment.
Nahi. Is the question clear?
We are counting the number of neighbors. Neighbors by which we mean Hamming distance is 1 for the two nodes.
Correct.
3 to the power n?
Still not correct, but you are headed in the right direction.
Then L to the power L, maybe.
No. So Hamming distance 1 means that you are different from this. So let me say I am that node and I am a sequence which is all A's. Okay. So my sequence is A, and it's L in length. Now my neighbor, the neighbor from me, which has a Hamming distance equal to 1, could differ L to the power 3. No, again, Akansha, but I think you are headed in the right direction. Let's see.
So 3L?
3L, absolutely, exactly. Right. 3L. Because think of it like this, that I am all A's. One of my neighbors would be that guy which has a different nucleotide from me at position 1, right? But if that guy has a different nucleotide at position 1, and because Hamming distance is 1, from position 2 to position L, it would have to be the same sequence as me, because otherwise he or she is not my neighbor, right? So how many sequences are possible which are different from me at position 1? Three, right? So at position 1, I have 3 neighbors. Now let me move to position 2. How many sequences are there which are different from me only at position 2? Again 3, right? And so on and so forth. Hence, for every position in the nucleotide chain, I have 3 neighbors who are different from me only at that particular position, and hence the number of neighbors that I have is 3L. Does that make sense?
Yes.
Yes, sir.
Anybody who's not so sure about it? Shreya, the problem that you made when you said 3 to the power L is that 3 to the power L is all the sequences which are different from me at all the positions.
Yeah, right, sir. I should have taken an or instead of an and.
Exactly. You did an and hence you get a much larger number of neighbors. Perfect. Cool.
Yes, sir.
Yeah, great. Anybody didn't make sense? Particularly the biologists? So everybody's okay with it?
Yes, sir.
All right, cool. So next question that I have written down is that why are we connecting nodes with Hij equal to 1 and not others? And the reason for that is that if I am a bacteria, let's think of this in context of bacteria. Wright certainly did not Sewall Wright certainly did not propose fitness landscapes thinking of bacteria. Wasn't even known bacteria have genes. He did it in context of eukaryotes, and we will look at another way to look at fitness landscapes as Wright proposed. But there is a reason I want us to look at landscape structures from the context of bacteria. And the reason is that all the experimental work that's done on landscapes in the last 10 years is done with bacteria, simply because it's at the state that we are in right now, it's only possible to do these with bacteria. And hence we want to understand landscapes in the context of asexually reproducing populations. If I am a bacteria with sequence all A's, right, and I reproduce, what is the most likely sequence of my progeny?
So can you repeat?
The question is that I am a bacteria and my sequence is all A's. If when I divide, what is the most likely sequence of my progeny?
All A's only. No?
All A's only, right? Because again, we discussed mutation rates last time, and we saw that from the context of bacteria, 999 divisions out of 1000 lead to an exact replica in terms of the DNA sequence that an individual is carrying. Does that make sense?
Yes, sir.
Right. So now let us think of this in terms of the graph that we made, the computer science graph that we made. I am a node which is all A's, and when I reproduce, my progeny is also the same node, more often than not, right, with a probability 0.999. What happens to the rest of 0.001 fraction of times? Geometrically, where will I be on that graph?
One of the neighbors.
One of the neighbors. Why not further away than that?
I think it's just the chances are extremely less that you'll have two mutations in the same.
Exactly. So the reason why in a fitness landscape we only join sequences of Hamming distance 1 is that when I divide, chances are that my progeny is going to be the exact same replica in the context of the sequence that the progeny is carrying, which means on that graph, I am at a particular node, my progeny will also be at the same node. Sometimes I will make an error in copying, and hence my progeny will have an error in it. And what that means is that it will be at a Hamming distance 1 from me, and that is why that progeny will be my neighbor. The chances that I make 2 errors while copying DNA are so small that it's almost impossible for me to have a progeny which is at a Hamming distance 2 from me, right? Hence, for all practical purposes, when I divide, my progeny either will be at the same node or will be at a node which is represented by Hamming distance 1 from me, and hence all the Hamming distance 1 guys are my neighbors. Does that make sense?
Yes, sir.
Any questions with that?
So this will mean that only at one place the sequence is different.
I'm not sure if I got your question, but if you're saying that Hamming distance 1 means that we are different in the sequence at only one place, yes. That is the definition of Hamming distance, right?
No, I mean that the chance of having mutation, which is very rare, can only occur at one, like to the most.
Well, no. What is the chance that 2 errors will happen, right? The chance of 2 errors happening is, see, 1 error happens in 1 in 1,000, right? So the chance of 1 error happening is 1 upon 1,000. Does that make sense?
Yes.
Chances of 2 errors happening are going to be of the order of 1 upon 1,000 whole square, which is 1 upon 10 power 6. Okay. Does that make sense?
Yes.
We'll do some of these numbers. I will show some basic calculations to illustrate that how unlikely is it for a individual bacteria to replicate and lead to an offspring which is at Hamming distance 2 from it. But for now, let's take it that by and large, it's going to be a rare event that you could lead to a progeny which is at Hamming distance 2 from you, and hence when you divide, you either generate a progeny which is represented by the same node or at a node which is at Hamming distance 1 from you, right? Is that clear, everybody?
Yes, sir.
The last bit. Actually, I do want to take this probably won't happen in 5 minutes. So let's stop here. The submission definitely gets pushed back by one class. So what I wrote, I don't know, 29th, I think was what I wrote. So it's not 29th now, it's due on 3rd of Feb, the 2 papers and their summary. But we'll continue this discussion on Wednesday. Unless anybody has questions about anything we did today, we can stop.
So,
Yeah.
in Hamming's distance, do we mean it is an amino acid or a locus, or what
No.
exactly do we mean when we
No, we just
place?
we're not thinking of anything like that. We are only thinking of DNA sequences. We don't care whether this is promoter region, where this is coding region, non-coding region, terminator, could be anything. Okay, we are only comparing DNA sequences and seeing where are they different, and if so, at how many positions, and that the number of places where they're different is the Hamming distance. Okay.
Okay, so it's nucleotide basically.
It's nucleotide, because when DNA is being replicated and errors are being made, it doesn't care whether this is coding region, I should make less errors, or anything of that sort. I mean, there are certain hotspots for mutations in the region, but we won't get into that.
Yeah.
Other questions? Okay, great. Let's stop for today then, and I will see you guys on Wednesday if there are no more questions. If you do have questions, feel free to leave. If you have questions, feel free to stay. We can discuss them for a few minutes.
Thank you, sir.
Bye. Akansha.
Yes, sir.
Can you switch on your video, please?
Okay.
We have a problem.
The recording is still on.
बंद कर देता हूँ.